Hi, everyone. I'm excited to kick off with this edition of Two Steps Forward, where we profile our founders who are pioneering the future. Today, we're joined by Maureen Hillenmeyer, founder and CEO of Hexagon Bio. Thank you so much for joining us, Maureen. To start, it'd be great to hear about your personal story and what led you to found Hexagon Bio.
I was always very excited about biology and medicine. I thought I would go to med school, but got really excited about research as kind of a bigger path or having a bigger impact on patients rather than one at a time, being able to discover cures for diseases, especially cancers, which I've been impacted by in my family. So after undergrad, I took a year off working in a lab at Notre Dame where I'd done undergrad doing research there. I knew how to code. This was in a malaria mosquito research lab around like 2002, 2003, when genomes were just starting to be sequenced more rapidly. So knowing how to code was pretty useful at the time, and not that many biologists knew how. So I started to see the application of computer science in biology and what a big impact it could have as more and more DNA sequences were coming out, genome sequences. So that's where the different pieces kind of came together. Like I could have a huge impact here, making discoveries that could impact a lot of patients if I applied some of these different techniques and especially the interface of biology and computer science, apply that to medicine. So that set me on the path of academic research. So I decided to do a PhD, came out to Stanford for a PhD in the program called Biomedical Informatics, which was just awesome. People working in the medical field, in the biological field, at the interface, and then computer science throughout. So let me pause there.
How did you discover the possibility of biomedical informatics as a field? And what led you to that program specifically?
Really, as the Human Genome Project was being done, and other genomes were starting to be sequenced. And before that, biology and computer science had been separate fields, but they were starting to merge around this time as all this genome data was coming out, DNA sequence data, and there weren't a lot of people that knew how to work at that interface, that knew both biology and computer science. And it was pretty clear pretty quickly to me what a big opportunity there was at that interface, like how much there was to be discovered. And it just wasn't moving fast enough because there weren't that many people working on it. So that's really what spurred me, like seeing this huge opportunity that wasn't being explored quickly enough or wasn't being developed quickly enough.
I know you referenced the desire to make an impact as a key driver in what led you to make the leap from scientist to founder. But I imagine you had some reservations as well. What gave you not only the conviction, but also the confidence to make that decision?
Well, I was running a group at Stanford working on the technology that later became and was licensed to Hexagon. So it wasn't actually so much of a leap. I was already kind of incubating this technology, working with people, some of whom went and worked at the company as well. So from that perspective, it was very natural. We were really developing this drug discovery technology initially at Stanford. And then I guess what really gave me the impetus or got me excited about launching a company instead was the scale at which companies can work versus academic groups. And the fact that everyone is really, like at a company, everyone's on the same team. I like to say rowing the same direction versus in academia. One of my old advisors used to say, in academia, credit is the currency, whereas in companies, money is the currency. And in drug discovery, that works really well because if we make good drugs, the investors that invest in the company do well. So incentives are aligned properly among a lot of the key stakeholders, investors, employees, founders. Whereas in academia, where credit is the currency, there's a little bit of different incentive. Everyone's trying to get their own paper. There can only be one first author on a paper. So there's lots of collaboration, but it's not exactly like everyone's on the same team in the same way they are at a company. So those pieces, the kind of being aligned, really rowing together on the same team, number one, and the kind of resources, the larger amount of scaling we could do, number two, were the reason I was so excited to launch a company.
I'd love to zoom out and think about the evolution of the problem space you're solving, applying innovation in DNA sequencing to discover future medicines. We've come a long way since the Human Genome Project you were referencing. It would be great if you can lend some insight into how this area has evolved and where you think it's headed.
So we work on drug discovery at Hexagon, especially oncology and infectious diseases. And historically, half of all cancer drugs were found from natural sources, and 3/4 of all antibiotics. So things like penicillin, Taxol, statins, these were found in microbes and plants. And the reason these evolved was over millions of years, these species in the wild evolved to compete with their neighbors out in the environment. So they evolved these really potent and specific drugs. Things like penicillin evolved in a fungus to kill bacteria. If you don't know the story of penicillin, you should read up on it. It's fascinating. And it was isolated by scientists many years ago for people who were overrun with bacterial infections in their own human bodies, and it works great. So that's a case that translated from the wild into human patients. It's similar story for Taxol, a cancer drug, and statins, a cholesterol drug. These things evolved in the wild for one purpose, and they are useful in human patients when something's out of whack that's sort of analogous to what evolved in the wild, that can treat whatever's too high and bring it down. In the case of Taxol, it's when cells are growing too rapidly, certain types of cells, this drug slows that down. In the case of statins and cholesterol, that's when cholesterol gets too high. There was a cholesterol drug that evolved in the wild because cholesterol is essential to cell membranes. So it evolved to knock it out kind of as a chemical warfare, just like penicillin, where these bugs are competing or killing each other off. But that's useful when our cholesterol gets too high. This anti-cholesterol drug can bring it down. So there's many blockbuster drugs that have been found historically. They're all found before DNA sequencing. So what's really changed is DNA sequencing. And as that started to get easier, we at Hexagon rediscovered some of those old blockbuster drugs, those existing blockbuster drugs. We could see in brand new sequence data, oh, here's penicillin. We rediscovered it using some new DNA sequencing approaches. Here's Taxol, here's the statins. We can see the genes that encode those drugs in the genomes of these microbes out in the wild. And then as we were studying that, we could also see thousands and lots more potentially unstudied drugs encoded in genes that no one had looked at. So that really opened up a door of a huge opportunity in terms of molecules that evolved in the wild that could become useful for human patients the way some of those prior examples had been.
Oh, that's fascinating. Can you tell us more about how you're harnessing these innovations to build a comprehensive drug discovery engine?
So we have a platform and workflow. It starts with what we call sequencing the earth. There's we're starting with fungi, but there's also bacteria and plants. But just within fungi, there's an estimated 5 million species on the planet. Only 5,000 or so have been sequenced, so 0.1%. And that's where a lot of these blockbuster drugs were found historically. So we need to sequence the other 99.9%, or at least a big chunk of that to really find the useful compounds there. So we have a team that's sequencing samples around the US to start, and potentially eventually around the globe. So that is building a large proprietary database of genomes, of DNA sequences. And that is a ton of data. So next we have data scientists and software engineers harnessing that data, analyzing it to find these new compounds. And they do that with machine learning. So training on some old examples like the ones I just mentioned and others, training algorithms on those known true positive examples, and then turning the algorithm to look ahead on all this new genome data, new DNA sequence data, and predicting what are the good drugs within there. So we have a lot of ways of doing that, or the parameters there are pretty exciting and evolving over time as we get more data. It's sort of compounding, like the more data we get and the more examples we get, the better the learning gets. So anyway, the next step then is actually making those molecules in the lab. And for that step, and we've published papers on this, we essentially transplant the DNA from these wild species into baker's yeast, just beer and bread yeast. I like to give people a tour. You can see flasks shaking in the lab that are growing yeast, and it smells like beer or bread, like it's just regular yeast that we all know and love. But instead of fermenting ethanol, like a lot of breweries do, we are fermenting these new drugs, or originally like testing out on penicillin, like having it ferment penicillin, statins, Taxol, et cetera, and now having it ferment these new chemicals. Then after that, we go and test those chemicals or compounds like for anti-cancer activity, so against cancer cells, and for infectious disease activity, especially systemic fungal infections. So testing whether these compounds can treat and cure those two types of diseases. And then within there, we have further stratified, like cancer is no longer one disease, it's hundreds of diseases as we're getting a better understanding of cancer. So we work on genetically defined cancers, so subtypes of cancer that are driven by certain cancer genes or oncogenes.
Thinking more broadly about your business outlook, I would love to get your take on what OpenAI represents for the world of big data. You have such a unique and vast proprietary data set. How do you think the ability to more efficiently combine different types of data will yield new insight in tech bio?
I am pretty excited about it. It's still early, but proteins and molecules can kind of be treated as images in some ways. And so there's already been some advancements in that kind of generative AI space on molecular biology. So AlphaFold, that wasn't OpenAI, but the AlphaFold advancements over the last few years have been pretty interesting. They're not like a panacea to solve all of drug discovery. Like a lot of human proteins that we need drugs for, there's already a crystal structure, like a solved, known 3D structure for it. But AlphaFold has been useful in other ways, like for us, working with these microbial proteins where there is no known structure. AlphaFold has been great and already had an impact on us. But then looking ahead to some of the OpenAI advancements and related advancements recently in language and images, it's kind of blowing my mind just the rate of change that things have happened over the last year or so. So if that trajectory continues, and if it translates to biology, so it's two big ifs, but then I think we could really see some big advancements in AI and drug discovery, where algorithms can predict molecules that could bind and block certain like cancer driving proteins or other disease proteins. So we're not there yet, but I am pretty excited about what's to come in the coming years. And then Hexagon is very nicely placed there because a lot of what the algorithms are going to predict are going to be really complex molecules that chemists can't make easily, like by traditional synthetic chemistry. So at Hexagon, we have all these biological machines that can make much more complex chemicals. I didn't explain that at the beginning, but those examples like penicillin, Taxol, statins, those are pretty complex chemicals compared to what traditional chemistry is able to make. And that's because there's these kind of biomolecular machines that are making them. So anyway, like fast forwarding, I don't know if this is like a 5-year vision or something, but if AI is getting better at predicting molecules, but then chemistry fails to make them, our platform could really easily step in. So keeping an eye on that for sure.
Incredible. And in closing, do you have any upcoming product news, open roles, or anything else that you'd like to share for those following Hexagon Bio's journey?
We also just closed a Series B. We'll announce that shortly as well. So we are hiring, and we have a lot of roles to fill. There's some on the website now. So yeah, this would be a great time to highlight that. We are growing. So anyone listening, it would be great if you could check out our website, see what open roles are there. And we're always looking for star scientists at kind of any level and position. So come join us.
Amazing. Thank you so much for joining us, Maureen, and to anyone out there who's listening.